Okay, hi everybody. I'm Stephanie Halene. I have the pleasure of introducing today's session of the RNA Collaborative. As first, I want to thank all the organizers for pulling this series together and running just a fantastic series for over a year. And I'm stepping in briefly for Karla Neugebauer, who will join us later to moderate. And it is my absolute pleasure to remind you to use the question and answer function to post your questions so that they can be seen by all participants and the speakers and hosts. And it is my pleasure to introduce Josien van Wolfswinkel, who will be our first speaker. Josien is assistant professor in the Department of Molecular and Cellular and Developmental Biology at Yale University. She obtained her PhD in chemistry in the labs of Ronald Plasterk, Albert Hegg, René Ketting at University of Utrecht on functional and mechanistic aspects of endogenous RNA and C. elegans. She performed a postdoc at the Whitehead Institute for Biomedical Research at MIT in Cambridge on link RNAs and planarian regeneration under the mentorship of Peter Redin, and joined Yale as assistant professor in 2016. She has been awarded several honors and awards, the Best Thesis Award, the Rubicon Fellowship from the Netherlands Organization for Scientific Research, the Vallée Scholar Award, and others. And today she will enlighten us about PIWI and cell function in planarians. So welcome, Josien.
Thank you so much. Thank you for that very nice introduction. I'm really excited to be here today and to tell you about the work in my lab. I can say briefly, I think we were interested in the RNA biology that underlies developmental phenomena. And one of the most fundamental developmental processes, I think, is the regulation of cellular identity. Most organisms start off their life as a single fertilized cell or zygote that has certain very specific characteristics. And that cell will divide, make daughter cells, and eventually those other cells will make all the cells of the body. So they will change their identity, they will differentiate. Some of them will turn into maybe neurons, or some of them will turn into muscle cells or epithelial cells. And these differentiated cells at that point look nothing like the original zygote, and also nothing like each other, both in their morphology and in the physiological processes that take place in them. Still, all these cells share the exact same genome with the exact same roughly 20,000 genes in there. And so what distinguishes these cells is which subset of these genes they're activating. During that process of differentiation they've gone through, they switch on certain genes, they switch off other genes. And so we can say that these transcriptional profiles that are changing over time is what identifies these cells. And these transcriptional profiles have to be stable in time so that the cell retains its identity during its own life. In many cases, it's even retained during cell division when there's other cells produced. But of course, they cannot be locked in place because the cells have to respond to other cues, and in some cases still have to be able to make other cell types. So that's it is clear that the regulation of cell identity is actually fundamentally one of gene regulation, and that gene regulation has to be very tightly controlled in cells in order for them to function. And gene regulation, interestingly, is an area where RNA biology is very important. There's lots of ability for non-coding RNAs to affect gene regulation because they have a very unique set of features that makes them very powerful molecules in these processes. RNA has a sequence, a nucleotide sequence, that allows them to really uniquely identify either certain transcripts or certain genomic regions, other nucleic acids, by base pairing. And when they find such a target, RNAs are very flexible molecules that can fold and recruit proteins, and that allows them to recruit very different functions to these targets. And so they can affect splicing, they can affect editing, they can affect the stability of RNAs, they can affect the translation efficiency, can sequester them into specific granules. In some cases, they may be able to recruit proteins to chromatin and change the organization of chromatin. So they are very versatile in what they can bring about. And in addition to that, they're also very dynamic. They can be very quickly synthesized and very rapidly degraded. And so together that makes non-coding RNAs very interesting molecules in gene regulation because they have this combination of specificity, versatility, and dynamic that make them very, very powerful. And one of the cell types, of course, that is very interesting in terms of development in this gene regulation is stem cells. These are the cells that can still divide, can make different identities. And so my lab is very interested in the RNA regulation in stem cells. And the model that we use for this is the planarian, Schmittea mediterranea, that is shown here. These are freshwater animals that live in almost any freshwater stream you'll find. These specific ones are from around the Mediterranean Sea, but you can find them in almost any freshwater stream. And they're very cute to look at. But what is more important to us is that they have a really large population of stem cells. That is shown here by two different labelings. One labeling cells in M phase, so in mitosis, another labeling cells that are going through S phase that are synthesizing the DNA. And so you can see that this is a really large population of cells in these animals. It takes up about even 25% of the total cells in the animal. And they're also distributed widely through the entire body. These cells are very actively cycling. They're constantly producing new cells. This allows us to, well, label them, to see the products of their differentiation. We can also use this to isolate these cells. We can visualize them. And because there's so many of them, we can even do biochemistry on them. So it's a very powerful model for us to look at the regulation of stem cells. And this constant cycling of the stem cells and constantly synthesizing new cells is necessary for them to constantly synthesize all the cells of their body. And even though they may look very simple, they actually have a lot of different tissues that they have to be constantly making. They have an intestine, a branched intestine, as shown here in green, where they digest their food. They have a pharynx, which is a muscular structure for which they ingest and secrete that food. It's connecting to the intestine. They have a nervous system that has a centralized brain region, ventral nerve cords. They have simple kidney-like structures, protonephridia. They have, of course, muscle that surrounds both the intestine and the body wall. They have epithelium. So all of these different cell types are constantly being generated, and there's a constant stream of all these cells being made. Not only are they doing this to replace tissues, these stem cells also allow them to recover from almost any injury. And when I say any injury, it really means even a decapitation, which in most animals is not survivable. For planarians, it's not a big deal. As you can see here, we can cut these animals in two pieces. They will continue to live, they will crawl away, and they will actually eventually regenerate and make both pieces will end up being a whole worm again. And the way they do that is that they synthesize this new tissue that is initially impigmented, that is newly synthesized cells from their stem cells. We call the blastema. And within that tissue, they resynthesize everything that is lost. So in the case that they have lost their tail, they will resynthesize the tail, make a new pharynx. In the case they've lost their heads, they will make a new head with a new brain, new eyes, connect everything back together, and they're good to go again. And they do this not once, they can do this again and again and again, seemingly indefinitely. And so the animals also, for this reason, they live seemingly indefinitely. They live forever, they don't age. And actually, to make it even more interesting, they also don't seem to generate any tumors or get cancer. So it's clear that planarians have very good control over their stem cells. They can make anything out of it. They can do anything with it. And so one of the core questions in my lab is, how do planarians do this? How do they keep such good control over their stem cells? They can constantly make anything, exactly what is needed, when it is needed, and nothing more, nothing less. So to understand how they're able to do that, we first got some inspiration from looking at other organisms that similarly are pretty good at regenerating and maintaining stem cells long term. And so when we look through the animal kingdom, there's actually many other species that are good at regenerating. Planarians are found here, the helminths, but there's also aceals and cnidarians that are very good at regenerating and living long term. And what all these animals share is that in their stem cells, they have PIWI proteins. So what are PIWI proteins? Well, they are these small non-coding RNA binding proteins. The small RNAs that they bind are called piRNAs, or PIWI interacting RNAs. And these are typically around 30 nucleotides in length, and there is a huge variety of them in an organism. They can recognize a target by sequence complementarity and regulate that target. And they can do that by recognizing an RNA and degrading it with the nuclease domain. In some cases, they can also translocate to the nucleus and affect chromatin state of a targeted locus. And one of their prime targeted regions, targeted sequences, are transposons. And these are these sequences in the DNA that can autonomously or non-autonomously, but can copy themselves, excise themselves, go to different locations, and can multiply in the genome. And so PIWI proteins are very well known for this in animal germlines, but they're also widely conserved in stem cells, in pluripotent stem cells, in animals that are able to regenerate. So when we looked at the PIWI proteins in Schmittia mediterranea, it turns out there's 3 different PIWI proteins: SMETWI1, SMETWI2, and SMETWI3. And first thing we did, of course, is to knock them out and see what this does to the organism. And you can see here that SMETWI1 knockouts don't do anything really. The animals live fine. But when we score how long the animals live when they start to die, SMETWI2 and SMETWI3 both are essential for viability. SMETWI2 is the most essential of them. The animals die really rapidly, even sooner than if we would take away their stem cells entirely. And of course, the first thing we thought is all of these genes are actually expressed in the stem cells. So I thought probably there's something wrong with their stem cells, but it turns out that's not the case. Actually, in the SMETWI2 knockouts, RNA of the expression of stem cell, typical stem cell genes is not altered, and the typical targets of them, repetitive elements, transposons, are also not really significantly changed. So the question is, why do these animals still die then? So to do that, we first synthesized an antibody against SMETWI2 to look a bit more clearly at where the protein is. And so while the RNA is present in the stem cells, and is almost uniquely present in the stem cells, it turns out the protein is actually more widely distributed. The protein is present, shown here in magenta. It's present in neoblasts. So here in green is a marker for the neoblast, and we can see that it overlaps in those cells. But it is also present in, for example, intestinal tissue. So in fact, it's present in every cell in the planarian body. And the other thing we can see from the antibody is that this protein is nuclear. It is in the nucleus of every single cell. So this explains why there can be a phenotype, even though the stem cells themselves seem to be perfectly fine. But then what does SMETWI2 target? Well, it is a PIWI protein, and so it's targeting something by its bound piRNA. So we isolated the piRNAs of SMETWI2 that are bound to SMETWI2 and asked, what do these piRNAs match to? And we see that a vast majority of them, or a large fraction of them, I should say, match transposons. And this is a very well-known target for PIWI proteins, so it made perfect sense to us. What was a little surprising is that there was a large fraction that we didn't see as matching transposons. It seemed to match other regions in the genome. Of course, we wanted to know what those regions were. And when we did that and mapped them to the genome, we found that actually these are regions that are in exactly the same places where the transposon mapping regions are. So these are just actually parts of transposons that are not well annotated, or little bits between transposons. But in conclusion, we can say that actually the vast majority of these transposons or these piRNAs matches repetitive elements. So we wanted to know what the effects of these this matching, what the effects of that was on the DNA. And so this being a nuclear protein, we looked at the chromatin organization. So we used ATAC-seq to look at the loci that are targeted by piRNAs and ask what is happening to them when we take away SMETWI2. And so this is what we saw. We saw here is shown on the x-axis the density of piRNAs. So more towards the right are the more highly mapping regions that have a lot of piRNAs. To the left are regions with very few piRNAs. And on the y-axis is shown the fold change in accessibility of the chromatin. And we see that there's a lot of regions that actually are not changing accessibility at all, no matter whether they're mapping to piRNAs or not. But in general, if we look at the overall trend, regions that have higher density of piRNAs tend to open up if they are changing at all. And the other thing we see is that a large fraction of those regions actually matches transposons. And so this fits with what we're seeing. On the other hand, there's also here a region or a group of loci, as you can see, that have very low piRNA mapping and seem to be closing down. And I'll get back to those later. So go back to the regions that are mapping to transposons. So now we're seeing that actually transposons are opening up. But in our sequencing data, we hadn't really seen transposons as being dramatically changed. So what we decided to do is look more specifically at individual transposons by staining and ask what can we see transposons being activated in these animals. And what we saw is that when we knock down SMED2, we do see various transposons being expressed. We see very little signal in controls, and when we do an RNA fish, so look at the RNA levels of these transposons, we do see clear signal in the SMED2. But what was much more remarkable actually is that these signals are very diverse, that each transposon seems to have its own unique pattern of expression. And these patterns resemble those of tissues. This pattern resembles the brain area, the brain lobes. This one resembles the intestinal structure, the branches, and these resemble some subepidermal cells. And so it appeared that transposons had very specific tissues that they were vulnerable in, that they could get expressed in. So I want to look at that a little bit more, or less in a less biased way. And so we isolated a bunch of different tissues and sequenced what transposons are getting activated in the absence of SMED2. And I found indeed that each tissue has its own unique set of transposons that are vulnerable in that tissue. So that was somewhat surprising. And we wondered, why is it that these transposons are activated in different tissues? We can totally imagine that some transposons never activate and that maybe some cell types are kind of insensitive, but we'd expected the same transposon to get activated to do that everywhere. So the first thing we thought, maybe there's actually tissue-specific transposons there, or tissue-specific piRNAs, and so that in each tissue, different transposons can get expressed. So we used the same strategy. We isolated these different tissues, and now we sequenced their piRNAs. And what I'm showing you here is the density of piRNAs to different transposons. Each dot is a transposon, and the dots are colored by whether the transposons are uniquely expressed in the intestine or uniquely expressed in the epidermis. And as you can see, it makes no difference for the piRNAs at all. So whether a transposon gets expressed in the intestine or the epidermis does not depend on the density of piRNAs in each of these tissues. There's the exact same number of piRNAs, and still it gets expressed in the intestine, but not in epidermis. So then why are these activated transposons different between the different cell types? Well, of course, the other thing that's very different between different cell types is their differentiation, the gene expression. And so we looked at these different cell types and asked what is going on in terms of differentiation, what is going on in the chromatin state. So we looked, what are the regions that open up either during differentiation to brain or differentiation to epidermis. And we found that there's unique regions, of course, in each of these tissues that are opening up or closing down. And then when we looked at what transposons are being expressed in each of these tissues, we find that they are highly enriched in the regions that are opening up specifically in that tissue. And because it's different regions in each of the tissues that are opening up, these contain different transposons. And this explains how there's different patterns of transposons that are being activated in the different tissues. And what we found is that actually this is not only a requirement for the transposons. Not only are transposons being expressed in these tissues when SMED2 is not there, we found that this also affects the differentiation of the cells themselves. So here we see a control tissue and a control part of an intestinal branch. We see nice staining of the intestinal tissue with an intestinal marker and very little transposon. When we now take away SMED2, transposons get expressed. But what in these same cells now, the intestinal marker is also not expressed anymore. So these cells are expressing transposons, but are not expressing the correct differentiation marker. And this leads to cells that are still there. They're still forming the branches, but they're no longer correctly differentiated. So what we see is that actually all the cells contain this wide repertoire of SMED2 proteins with bound piRNAs, with a wide repertoire of piRNAs for all the transposons basically that are there in the genome. And when these cells start to differentiate, they will open up certain region of their chromatin and certain transposons will be vulnerable. So for example, when they start to express this blue transcript, this orange transposon region might become accessible and could get expressed in that tissue, except that there's a SMED2 protein with a piRNA ready to attack it and bring it back under control. So planarians actually use these PIWI proteins as constant chaperones to control their genome. So I want to take a few minutes still to talk about another project, and I realize that it's already quite a bit, but you may wonder what happens with these other PIWI proteins. So there's 3 of them. And one we are particularly interested in is SMED1. It's a very close relative to SMED2 that I talked about just before, but it's a very different one in the sense that it doesn't seem to have any phenotype. And the reason we know SMED1 so well is that it is the marker of neoblasts. It is the marker we always use to label the neoblasts, to label the stem cells. It's a perfect staining in the RNA level. And even on the protein level, the protein is actually present exactly in the neoblasts, and it stays around a little bit longer after that, but quickly disappears. So we use this constantly as a marker, yet there appear to be no phenotype when we knock it out. So we decided to just push this experiment a little bit further. And instead of doing RNAi, it's typically done for about a month or so, we decided to just keep going. We found that if we do that, actually a phenotype does appear. It takes about 3 months of RNAi, but it leads to a very dramatic phenotype where these animals are completely unable to regenerate. So whereas a control animal after amputation forms a nice blastema, the SMED1 knockdown animals form either nothing or kind of an open wound that doesn't really close. And this phenotype gets increasingly strong over time. Again, this protein is very highly expressed in the neoblast and not much else. So of course we wanted to see what the effect was in the neoblast, and the answer is almost nothing. All other genes seem to be perfectly nicely expressed. Even the mitotic response, like the more mitosis that typically happens after wounding, is perfectly normal in these animals. So they are still making new cells, even though they're unable to really do anything with it. And we looked at it in many other ways. It turns out initially the wound response, anything to that is perfectly fine until after about 3 days, they just get stuck and they don't get any further. So we want to see what are genes that are being changed or that are changed in these animals. And for that, we did, of course, RNA sequencing, and then we compared the genes that are changing to single cell RNA sequencing data where we had grouped genes into what tissues they correspond to. And here you see here is a simplified Seurat plot showing different cells and showing the different expression patterns. So we see neoblasts, 2 different types of neurons. We have the muscle, intestine, epidermis, and catapsin cells. And so when we asked the genes that are changing, what kind of gene or tissue class do they belong to? We found that overwhelmingly, these are late epidermal genes. And we were actually able to confirm that by doing a staining in these animals and see that that's indeed muscle cells seem to be fine, muscle structure seems to be fine. Mature epidermis is even pretty okay, but it is this late epidermal stage where we see a dramatic difference between control animals and SMED1 knockdowns. And this is kind of, well, the epidermal lineage is a good one that we know quite a bit about it. We know that there's a lineage of differentiation that's happening, and we know a bunch of genes in all of these stages. And especially this late epidermis stage is one where a lot of the genes that are being expressed signify that these cells are factories. They're making stuff. There are a lot of mitochondrial genes, a lot of mitochondrial activation, and a lot of proteins that we don't know quite what they are because they don't resemble anything, but they're being made in really high amounts. And so after a bunch of experiments that's looking into what could possibly be wrong here, what we found is that what is wrong in these animals is their extracellular matrix. So what these cells appear to be making is this extracellular matrix shown here in yellow that attaches the epidermis to the animals. In the SMED1 animals, this ECM is very thin and the epidermis is not well attached. And whereas in a control wound, we can see this kind of spider web of extracellular matrix being formed to kind of get the wound back together. In the SMED1 animals, that doesn't happen. So that's great. Now we understand why they're not regenerating. But the question is, how does this relate to SMED1 and piRNAs? Of course, we again looked at what are the small RNAs that are being bound. And we find largely the same thing as with SMED2. We find that our overwhelming majority is related to repetitive elements. The repetitive elements themselves don't change much. And that is not so surprising, because we knew already that SMED2 is there in the nucleus to keep control of everything. In fact, we find that if we take away SMED2, now we do see that transposons are being expressed. So here we see a group of different stem cells, and in magenta are 2 different transposons. In the controls, the transposons are not expressed. Actually, in SMED2, the transposons are not expressed in the stem cells because SMED1 is there keeping control over it. When we take away SMED1 as well, now the transposons are expressed. So we see that SMED1 actually is a great backup filter for transposon transcripts. But that can hardly explain why there is a phenotype without SMED2. We also looked at what genes are expressed or are altered. And in general, again, transcripts that have a lot of piRNAs are not being altered. In fact, even we looked at this epidermal transcripts, and they have very, very few piRNAs mapping. So if anything, they're even further depleted. There's some transcripts, though, as you can see here, that do have piRNAs and do alter. And those turn out to be largely histones. And interestingly, they're not all histones. So when we looked at them in more detail, we realized that the genes that have the largest fold changes are histones that we thought were lowest expressed or lowest in their expression levels. And we looked at it even further. We realized that this is because histones come in 2 flavors. There's canonical histones that have stem loops, their terminals, and histone variants that have 8 hills. And these 8 hill ones are much more abundant in our sequencing because we often do oligo-DT selection. But the canonical ones are the ones that have the piRNAs and that are changing in SMED1 RNAi. So these are transcripts that are actually a little odd. They don't have poly-8 hill. And that turns out to be a little bit of a theme in SMED1. So we see also that there is actually, even though it's not huge, a larger fraction of structural RNA that is found in SMED1. And we see that certain transcripts, for example, the SNRP U2, accumulates in these cells. And there are several other transcripts, similar like ribosomal RNAs and new RNAs that are higher than expected in these transcripts. So what we're finding is that out of this universe of RNA that is there in the stem cells, SMED1 seems to be mostly targeting things that look a little bit off and is keeping them under control. Whereas transcripts that have nice poly-8 hills are not regulated by SMED1. And in particular, these epidermal ones turn out to be essential to keep active. And when SMED1 is not there, they seem to be not protected anymore. And interestingly, we're finding that there's also a poly-A binding protein specifically in these epidermal cells that might help explain why this is so important. So with that, I really want to close this down and say that so we're finding that PIWI proteins are hugely important for stem cells, and that they protect both the genome and the transcriptome. SMED2 protects the genome and keeps the things silenced that should be silenced, and SMED1 is the one that protects the transcriptome and keeps the odd-looking transcripts out and the good-looking transcripts active and there. And with that, I'd like to thank all the people in my lab who've been working on this, especially Danyan, who's been working on the SMED2 project by herself, and Michael and Danyan and Krishna who've all been working on SMED1. And thank you very much for your attention.
Thank you, Josien. Hi, Karla. Welcome. I don't know if you're able to join. Maybe if not, or while you, oh, there you are.
I can unmute myself, but I don't know if I'm on video. Am I?
Not yet. Oh, there you are.
Sure.
Okay, I don't see myself. I think there are some questions. I got a chat from Liz that there are some questions in the chat.
They're in the Q&A.
The Q&A, I see. And shall I ask them then? Oh, Bill Marslove. Hi, Bill. So Bill is saying, as someone who works on histone mRNAs, is the regulation by piRNAs post-transcriptional?
Yeah, that's a very good question. I think it will be, because we don't see effects on histones on the chromatin level. We don't see changes on the chromatin accessibility. We see this only actually in SMED1, which is the PIWI protein that is not nuclear and that's in the cytoplasm. So I think that is indeed what is going on.
There's a second question from Kangyan Zhang. What about the function of PIWI3?
Yeah, PIWI3 is, so there's a whole thing in the piRNA field what we call the ping pong, how piRNAs are going back and forth and amplifying. So PIWI3 is on the other side of SMED1 and 2. And so it's the other side. It seems much more promiscuous in what it takes up. And to be honest, we don't quite know what it's doing at this point.
Good answer. Let's just go for the last question and try to answer it briefly, because it's 4:30 and we want to get started with Stephanie's talk. But Astrid Haza is asking, do you think that piRNA biogenesis is regulating the level, these structural RNAs, like an innate response, or do you think that these piRNAs have an adaptive function? And she's congratulating you on your fabulous results.
Thanks. So yeah, I think it is regulating, maybe indeed like an innate response. Maybe it's seeing something about these RNAs that's a little off. And if whenever they are trying to, well, get out of the nucleus or do things where they shouldn't go, that's SMED1 is controlling their levels. But clearly it's important for the cells to keep doing that, to, well, maintain their stability over time.
Cool. Okay, so thanks everyone for your questions, and thank you, Josien, for your wonderful talk, some of which I missed. I apologize. I don't know if Stephanie explained that I had a dental procedure that I couldn't change. So fortunately, my face isn't too disfigured that I can't introduce Stephanie. But I'm so proud of Josien and Stephanie, really active members of our RNA community here at Yale. So I'm especially pleased to be able to make it here today. So maybe without further ado, I'll say a few words about Stephanie, who's our Chief of Hematology Division here at Yale. She's a wonderful collaborator, and what I really admire about her research is that she uses cutting edge basic RNA biology methods to really understand what's going on in the context of hematological diseases, in particular MDS and AML. And I think you'll appreciate that from her talk, so I won't go on in excruciating detail. But to say that she's a devoted clinician and also a devoted basic scientist is fair enough. She got her MD and her PhD in Germany at the Eberhard Karls Universität. And then she did her postdoc and her residency in LA. I'm not exactly sure how she found her way to Yale, but I'm sure glad she did. So she's been here since the early 2000s. And it's just really wonderful to have a clinician who's so conversant with everyone in our biomedical community. And so I'm really grateful for her contributions and can't wait for you to hear her talk. So I'll be quiet now.
Thank you, Karla, for the kind introduction. And I also want to thank Liz, Stacy, and Grace for organizing today's seminar. Okay, let me share my screen. Okay, and minimize this. Okay, next slide. So let me get kind of right to the story. So my laboratory is interested in myelodysplastic syndromes and acute myeloid leukemia and also hematopoiesis in general. This talk is really about MDS and a subset, a secondary AML. So MDS AML, they're common, relatively common, way over 20,000 new diagnoses in the US per year. They're caused by a variety of aberrations in a variety of pathways, genetic, epigenetic, transcription, signaling, and obviously important for today's talk, and splicing. And what you can see here is that, MDS AML occur in, with aging. but you can also see that the survival is not as good as we would like it to be. And on the right side, what you can see here is that mutations in splicing factors are actually found in over 50% of patients with myelodysplasia. And this shows you that these are not some distant splicing factors, but they're really factors that are part of the spliceosome, and they're really required for basic splicing. And they occur in 50% of the patients. When we look for these mutations, their allele frequency is high, 40 to 50%, which tells you that these mutations are not passenger mutations. They occur in the dominant clone. They're also mutually exclusive. So really one patient, one cell has one mutation. More mutations are not tolerated. And then of course they occur with other known mutations and in certain diseases, certain subtypes. So there's also some disease, some phenotypic specificity. And so they are clearly driver mutations. And this talk today is about U2AF1 or U2AF35, which is generally part of the U2F1, U2F2 heterodimer, and really important for splicing at the 3' splice site. And the mutations in S34 and Q157 occur in these 2 zinc finger domains that have been shown to contact the RNA directly. And so when we embarked on this project, we asked several questions. And who is we? So that is Giulia Biancon, a super talented postdoc in the lab, and then Toma Tebaldi, who now has his own bioinformatics and wet bench lab at the University of Trento in Italy. And they've done this work with a great team, but they've really led this work with their spectacular skills and knowledge. So the questions we asked are, well, how do U2F1 mutations affect RNA binding in vivo? How do mutations then alter the binding splicing relationship? And what are the biological consequences that ultimately contribute to the disease process? And, oops, my apologies. Okay, I cannot move backwards, but obviously these mutations can do a whole bunch of things on splicing, binding and splicing, but can we also discover the, the biologic function? And so to answer these questions, Giulia developed several techniques. One is fractionated eClip. I'll show you the details. She then integrated eClip and RNA sequencing, and then also used immunofluorescence imaging. Oh, I think I'm on a timer by accident. And time-lapse-seq and single-cell RNA-seq to solidify the results. And for this, she generated a cellular system where she endogenously expressed flag, tagged, wild type, and mutant U2AF1. And she also verified her results in, oh my goodness, I do not know how to stop the timing. Do you mind if I briefly unshare?
Yeah, go for it.
I don't know how to stop the timer. You know what? Let me do this.
Yeah,
let me share my screen and
yeah,
just not go in presentation mode.
Exactly.
I apologize. Okay, so she used
that looks good.
How's that?
Yeah. Maybe close the right-hand panel. Yeah.
Yeah. Okay. All right. Thank you. And sorry about that. Okay. So she used the kind of exogenous expression cell system, and then also validated the results in CRISPR-Cas9 modified cells, so where the endogenous gene is modified. And she also used primary cells from MDS and AML patients. And so this shows you the eClip protocol, where you cross-link the proteins to the RNA in vivo. You then digest the RNAs to obtain these fragments that are protected by the bound protein. You immunoprecipitate the protein of interest with specific antibodies. The RNA protein complexes, or the RNAs, then labeled with P32, run on an SDS page, gelled, and the RNA protein complexes are visualized by the P32 attached to the RNA. And then the RNAs isolated, reverse transcribed, and then eventually sequenced. And how do you then read this data? So the reverse transcription kind of occurs from the right to the left here. And at these cross-link sites, the reverse transcriptase falls off, and that is then the end of the read. So the end of the read +1 gives you an idea where this protein was cross-linked or directly touching essentially the RNA. And what I'm showing you here is right here's this the 3' splice site. So shouldn't we just get reads that are at the site? But what we quickly realized is that UTF1, UTF2 are kind of in a tight, I don't know, complex or close together. And so we expected that we would potentially also see UTF2 binding because UTF1 and 2 are kind of connected with this RNA read. And when you look at the gel, you can see that this RNA smear kind of extends right kind of above the size of UTF1, which is around 35 kilodalton. UTF2 is around 65 kilodalton. And so to distinguish UTF1 binding alone from potentially binding of the two proteins, Julia fractionated the gel and isolated independently RNA between 35, 65, and then 65 and above. And this is what you can see here. So these are now, so these dots here all are essentially ends of reads and cross-link sites. And what you can clearly see is that UTF1 binds on this A at 3' splice site. And then when she looks at the heavy fraction, you can see that we're now picking up what is very, very likely or surely the UTF2 peak along the polypyrimidine tract. And so now in the next step, okay, the animations don't work, but just don't worry about that now. So in the next step, she can then look at how do the mutations now alter RNA binding? And in gray, you see the wild type. In orange, you can see the S34F mutant, and you can see that it picks up an additional peak at this -3 position. And the Q157R mutant picks up an additional peak at this +1. And then you can see here, again, the heavy fraction that there seem also to be some changes on the of UTF2 binding. So now how does this correlate with splicing changes? So these data had been also previously published by other groups, but you can see that splicing changes in the S34F mutants really are dictated by the sequence at the -3 site. So if there's a U, there's more exon exclusion, and the splicing changes in Q157 mutants are dictated by this +1 site. So these binding peaks that are induced by the mutants also determine whether the splicing, this sequence-dependent splicing changes. And so when she then looked at, okay, how does binding specifically at certain junctions affect splicing? The question she was asking is, okay, if we have more binding, do we have more or less inclusion or exclusion at these junctions? And if it's directly proportional, you would say, okay, if you have more binding, you also have potentially more exon inclusion, and you have less binding, you have less exon inclusion. But and these would be some examples here. For example, an example of more binding of the mutant U2F1, and here's less binding of the mutant U2F1. And so you can analyze all these, all the junctions to determine what the predominant pattern is. And what you can see here that with the S34F mutant, the higher number of events seem to be with more U2AF1 binding, but less exon inclusion. And you can also see that in these reads. So you have more mutant UTF1 binding at this -3 peak, but it seems to actually affect binding of U2AF2, which seems to be reduced and which could be the cause of the decreased exon inclusion. And she depicted that in this graph, where maybe there is somehow a change or effect of U2F1 on how U2AF2 can contact the RNA or splicing kinetics. So we don't exactly know that. Of course, there are also other events, actually essentially in all quadrants, and that tells you about the complexity of binding and splicing, that there's not a clear one-way correlation. Similarly, she looked at this for the Q157R mutations, and there she gets a different picture where there seems to be less binding and less exclusion and less inclusion, and that's kind of shown here. So there is the mutant is in brown, so there's less binding here, and again there seems to be also less binding of the U2AF1 at these junctions. So that is the predominant type of relationship she finds in the Q157R mutation. And that is relevant because while these two mutations occur in the same gene, they do they are different in their phenotype, even in patients. So since Julia's cell system relies on exogenous expression of the flag tagged wild-type and mutant U2F1, she analyzed many data sets that have been splicing data sets that have been published, and then looked at how well her system represented the other systems. And so what she shows here is that there is an overlap between this HEL S34F data set and published data set in over 2,700 genes. And if she looks here at this CRISPR-Cas9 modified model, a gift from Rafael Bejar at UCSD, there's a very high overlap at the gene level, but there's also very high overlap at the junction level. And I think an important question is so that if there is inclusion, exon inclusion, for example, in one system, there's also exon inclusion in the other system. So there's very nice correlation, suggesting that we're not only capturing the binding splicing, but we may also be able to capture relevant biology. And so she then integrated the RNA binding splicing data and looked at what biological processes may be affected. And one aspect that struck her was that this kind of enrichment for biological processes of regulation of organelle assembly and then cytoplasmic stress granules. And this is the image you missed earlier because of the animation. So the question is, could stress granule biology play a role in the disease biology? So what are stress granules? They are very dynamic, membranous organelles between proteins and RNAs. And they are thought to improve cellular adaptation to stress by probably stopping unnecessary translation and allowing the cell to focus on survival. And other people have shown some role for stress granule formation in cancer and tumorigenesis. So let me, we have some time. So so the next thing Julia did, she then looked at whether known stress granule proteins and transcripts were actually enriched in a data set. And that's what you can see here. So she, from several IP and other data sets, she specifically looked now at stress granule enriched proteins or stress granule enriched or depleted transcripts. And these enriched proteins and transcripts are enriched in her data set also, which is shown by the fully colored circle, and the odds ratio is depicted by the size of the circle. Whereas, for example, in depleted transcripts are not enriched in her data set, so suggesting that there may be a biologic function. And so then the obvious next step for her was to look whether the wild type mutant cells, whether there was a difference in stress granule formation in the mutant cells comparing to wild type cells. So again, she used her mutated wild type S34F or Q157R inducible health cell system. Health cells are human erythroid leukemia. and then stained these cells. She then stressed these cells with sodium arsenide for 1 hour and stained these cells for G3BP1, which is stress granule marker, and then DAPI to look at the nucleus, and then performed confocal microscopy to very carefully look, at Z-stacks and localization of the stress granules. And what you can see here, these are U2F1 wild-type expressing cells stressed with arsenide, and these are U2F1 mutant S34F or Q157 expressing cells also stressed with arsenate, and you can clearly see in the quantitation that these mutant cells have enhanced stress granule formation. So that is a saline system, and it's an inducible saline system. So you always have to be concerned whether that really is biology that we may be seeing in patients. So she then took U2F1 wild type and S34F mutant primary cells from patients with AML, stressed these primary cells again with sodium arsenide, performed the same staining. And here you can see the same results, that mutant cells have enhanced stress granule formation, and that is quantitated here on the right side. Since RNA binding splicing, it could be secondary effects, for example, affecting transcription, or could it affect RNA stability? And so for that purpose, she, with Matt Simon's lab, with Josh in Matt Simon's lab, performed time-lapse sequencing, where newly transcribed RNA is labeled through mutation from U to C through thio for U incorporation. And then again, she looked at down versus upregulated transcripts, and to see whether there would be increase, whether the levels of the RNA would be affected by stability or synthesis. And what she found was that again, transcripts enriched in stress granules are particularly stabilized in S34F cells. And they're also, there's a trend that they're stabilized in Q157R mutant cells, and whereas depleted transcripts are destabilized or shut down. So suggesting that stress granule enriched RNAs in mutant cells have higher higher stability, and in the Q157R mutant also some increase in synthesis. So as a next step, she actually did, I think, a very nifty assay. So she chose a U2F1 wild type MDS, a mutant MDS patient, and performed single cell RNA sequencing on these cells with a 5' methodologies, so that she could actually detect the S34 mutant at the RNA level, and then looked again at a stress granule expression score. And what you can see here is that right here you see the wild type single cells, and here you see the mutant single cells where she can detect the S34F mutation at the RNA level, whereas these are still cells from the patient with a mutation, but where she could not call the mutation. And what you can see is that clearly in the mutant cells, the stress granule expression score is enhanced. What I'm not showing you because of the animation is that there are also subclonal mutations in NRAS and KRAS, and there was a prior description that NRAS KRAS mutations may also enhance stress granule formation. And when she looks at the subset of cells with NRAS KRAS mutations, she can see an even further enhancement of the stress granule expression score, again, underlining that stress granule biology may actually play a role in the clonal advantage that these cells have in patients. And so then ultimately, to show that there may be this biologic function, she chose an inhibitor of this integrated stress response, ISRIP, which antagonizes the translational effects of EIF2 alpha, and essentially counteracts this enhanced stress granule response. And so what you can see here is that mutant cells have improved cell viability when stressed with arsenite. But when she now adds this inhibitor to the wild type or mutant cells, this enhanced viability is actually is lost, suggesting again that this enhanced stress granule formation may actually result in improved cell survival and potentially contribute to clonality in patients. So in summary, Julia and Thomas showed that mutant U2F1 binding, mutant mutations in U2F1 alter RNA binding. They alter RNA splicing and ultimately may contribute to enhanced stress response to result in clonal advantage of these mutant cells. And this work was done predominantly by Julia and Toma, but in collaboration with Matthew, Purval, Torben, and many others. And we have to thank our phenomenal collaborators. So Jörg Beversdorf's group, Matthew Simon's group, Karla's group, Hai van Dins work, and Gabriela Viero in Italy. And we made it through the pandemic with ice cream in the cold and more recently pizza. So thank you, everyone, and my apologies for the slides.
I feel like every time we go to do Zoom, there's some kind of technical problem and you just can't believe it because we've been doing this for so long, but there you go. Great talk, Stephanie. I've got 2 questions in the Q&A. What does that sound? Does anyone know? Okay. One of the questions is from Chao Yi Yang, and it's a simple question that's asking, does U2AF1 recruit U2AF2? And I think that's worth maybe discussing the assumptions about which protein's binding event recruits the other, and then whether that binding might be remodeled. I should let you answer that question.
Oh, I think that's a difficult question. And other people may know that better than I do. So I think one of the reasons why we really wanted to look at cells where the endogenous protein was mutated was because we definitely changed the stoichiometry, somewhat by expressing endogenous U2F1. And we have done some U2F2 eclipse on cells where we do or don't exogenous express U2F1, and we can actually see some changes in binding. But whether it's clearly one recruiting the other, I think that is, I don't think we can answer that just from these assays.
I guess I assume that U2AF2 is the dominant RNA binder, but then of course we know that U2AF1 is very importantly interacts with the 3' splice site. So you're very clearly showing what I think of as a remodeling event, but I appreciate the question because maybe we have to think about that more carefully, as a field, like how the U2AF, the 2 subunits interact with the 3' splice site. Okay, so Harry Yang is asking, is there a way to explain how the increased binding on S34F mutant on transcript leads to more exclusion of cassette exons?
Yeah, I think that's kind of the same million-dollar question that I think we can only speculate. And I may have been too, this is just how it is in the talk. So I think the fact that we're seeing, more binding and all sorts of, that it's not just one outcome. I think that's the complexity. And it's probably additional proteins at different junctions that come into play. And our eclipse, it's still a static, right? It's one moment where we cross-link. So the one thing we certainly can't say anything about is kinetics.
Right. Chao Yi Yang also had a question that was moved to the answered, but it's a body question. It's P-bodies store mRNA for degradation. or something like that, right? Is P-body biology affected by mutant UTAF1? So, to what extent have you checked other bodies, I guess, is part of the question. And he was wondering this because mutant UTAF1 should generate misspliced mRNAs, which might increase the burden of mRNA degradation. So,
yeah, Julia's very interested in the organelles. I think that's what she wants to take forward. We haven't looked at that.
I would expect the misspliced RNAs actually largely to be degraded in the nucleus, but it's true that certainly the NMD type, degradation would happen in the cytoplasm, but not in P-body. So yeah, that'll be interesting to see as an overall.
Oh, and I like to see Clara Kirchhoff's answer. UTF1 is an obligate heterodimer, but U2AF2 is not. And,
right. Yeah.
And that's the thing.
Clearly, but if Clara were here and we were having martinis, but clearly, even though it's a heterodimer, clearly where exactly it's interacting with the RNA is mutable because
I have to admit in other fora, whenever I'm asked, it's like, I have to ask that to Clara Kirchhoff because she is the expert.
Right. Right. Oh, so it does open some interesting questions. Okay, so there's another question from Mariela Cortez Lopez. Are the granule proteins differentially expressed in mutants, differentially distributed also in the different cell lineages, HSPCs versus EPs? Do you understand that question?
So Julia did some Western blots on a bunch of granule proteins in these cells, and she did not find differential expression of the proteins, but we haven't looked at different cell types, for example, in, stem cells at various stages of differentiation. So we haven't done that. So we definitely welcome any suggestion of
that's
how we should
that's actually a really interesting question.
Yeah.
Okay. Any further questions from the audience? I wish I could see. I don't know if anyone can. No one can raise hands, can they? So, yeah, these are really very fascinating questions. I'm rereading them as well. These kinds of results cause you to rethink a lot of things that you sort of assumed in the past. And I did have one comment from a member of the audience who liked the idea of having martinis at this event, but clearly, it's BYO. So, all right. So I guess it's up to me to thank both Stephanie and Josien for their amazing talks and the audience for their questions. So thank you, everyone, and please enjoy your evenings or the rest of your day, depending on your time zone.
Thank you, Karla. Thank you to everyone.
Bye-bye.